might just hold over any questions through to the panel discussion that's going to happen now. And so before Mark sits down, we might invite him back to take his seat along with Bjoern and Bruce and the surprise guest, Liset.
Behind here, yeah.
And so we might start off by directing any questions to Mark to get things rolling from his presentation. Any questions? Perhaps I can start. Mark, where do you think we should invest our energies?
Gosh, what a question. I guess we should invest our energies where we've seen most promise. And I think it's fair to say that the pattern recognition methods have shown some very high promise in that there does seem to be some good evidence that that approach works. However, it's also clear that that approach doesn't necessarily make best use of what we already know about epilepsy. So it may be, this comment may be biased by my own interests, but I suspect it would be profitable to look at how multi-scale models of epilepsy may inform the approach that we take to measuring and analysing signals in order to identify seizure predictive signals.
Okay, thank you. Any other questions of Mark? Yes, up the back.
It was great to say that how far shall we go, but maybe I'll give a little bit another context. How far do you think we should try to understand the operation of the neuronal system before focusing specifically on epilepsy? Because we know very little about, for example, how our brain makes very elementary calculations, and we start with one specific pathological condition. Shall we not also understand migraine along with epilepsy, or maybe post-ictal suppression or spreading depression, and then put epilepsy as one particular case? Or shall we focus exactly on epilepsy? How broad shall we approach the question?
Yeah, that's a very interesting approach. So what would I say to that? I guess that to a certain extent, or to a considerable extent, I don't particularly care whether a method is based on really in detail understanding underlying mechanisms. So if a seizure prediction method works, great, it works. So this kind of pattern recognition method, it works. And it doesn't rely on understanding the underlying system. And if you want to say, should we try to understand the whole breadth of paroxysmal brain conditions? Well, epilepsy is very common. Arguably, it's more disabling than things like migraine, and I would certainly put it right at the top of the priority list of brain conditions, paroxysmal brain conditions that one might treat in this way.
Okay, other questions? Yes.
I would like to ask you about your opinion about measurement other than EEG about prediction. Because we also discussed whether those like the heart rate or the glucose level or the lactic acid level are all of these are the result of the epilepsy or result of the epileptic discharge, or they are accompanying denominator, or maybe can they be the predictor of their decision?
Okay, so I'm sorry I didn't catch all of that, but I think I caught enough of it. Again, from a personal perspective, I think it's probably only worth pursuing other modalities, tracking other things other than EEG, if it's easy to do it, or easier to do it than EEG. So one of the reasons for just simply mentioning the changes in cardiac autonomic drive in association with seizures is that that's very easy to measure, relatively speaking, compared to EEG. So, one could imagine having some kind of cardiac monitor that is wearable every day, day in, day out, applied by the patient, whereas thinking that you could have some kind of EEG that a patient could apply every single day, we don't have that technology.
Graham.
We know that preceded by psychic symptoms. Is there any technological advance that would lend itself to being able to monitor over time, such as functional MRI or something of that nature, which would sort of pick up psychological or thought disturbance that might precede?
Yeah. Okay, so I guess you're talking about prodromes, the kinds of things that many patients probably retrospectively associate with predicting a seizure. I know that, I think it was from Andreas Schulze-Bonhager's group, they tried prospectively to log those kind of prodromal symptoms and see if they associated with seizure onset. And they didn't seem to associate with seizure onset. But whether one could track behavior in a way that might be more detailed than logging, I suspect you could. And certainly I know there are some moves afoot at the moment to set up long-term monitoring projects that might look at motor behavior, speech patterns, things of that kind that can be monitored automatically and analysed automatically. What that will show, I don't know. I think to a certain extent that's a kind of fishing expedition, but it'll be very interesting to see what comes out of that.
Brian.
Yeah, so one of the challenges, I see that I grapple with a lot is evidenced by, the selection of people who are sitting up in front of us. So I look at the work that Liset does, and I see like tremendous opportunity to bridge it to the kinds of clinical and animal studies that you showed in the breadth of your presentation. But I struggle with how we could actually start to get it using some of these mechanistic, cellular, physiologic kinds of experiments, but get them into people, because we have these very old animal models that we have a hard time understanding how they relate to some networks. So there's one of the things about this group that makes it so wonderful is we have communication people. We have people like Bill Stacy and Steve Schiff who are look at cellular models. We have people like Sid who are putting Utah rays into people. But I can't get at the circuit physiology. I'm so jealous of the Parkinson's work, like Malin DeLong's work, where he has a well-defined circuit that they can explore in a very animal model. How are we going to bridge this divide between the basic science of isolated circuits in animals and the human condition?
Yeah, this is a very important challenge indeed. And in my talk yesterday, I tried to make the point that if we really want to bridge these two perspectives of the problem, the experimental research and the clinical perspective, we need to make common routes to try to combine experiments or observations and to try to extract the information in a common sense. So in particular regarding this problem of trying to identify preictal event that can be useful for seizure prediction or at least a warning system, I think that it is imperative to divide the problem, to separate the problem in time and space. In time regarding whether a preictal state exists, whether this preictal state runs in different temporal scales, from minutes to seconds to milliseconds, and what different signals are running in each of these scales. Because we have been focusing, for instance, in electrophysiological recording, and probably they are informative just at the very last minute. And other neurovascular or different readouts are more related with a longer time scale. But then in the spatial case, and I think that this is an important, because in clinical setting you cannot choose where you will stick your electrodes. And in animal research, we know exactly what to do and where to go, even if we are making a mistake, or even if we started experiments with a wrong idea, we can correct over the course. But in human, you have to stick the electrodes guided by preoperative information, and therefore you need to try to make sense of the data that you have. So in that perspective, I think that recent concepts like the penumbra area, which are not recent concepts, but concepts that are brought to the problem, are useful, because the signals that you can record from the core and the signal that you can record from the surrounding territories, or the signal that you record from a projecting area, will not mean the same and cannot be interpreted in the same way. So for me, the challenge is to try to make some order on how we collect the data and how we compare the data. But yes, that's a major challenge.
So this is a huge problem on the clinical side.
Yeah.
Because even when people localize, there's so much variability from place to place,
Yeah.
and verification is an issue. So, I mean, this is a crusade of my own, but data sharing and transparency is something I think is huge and probably necessary.
Steve.
So, a question for the panel. Is there an analog? And what I mean by that, this was a major issue in the early 20th century for how to predict weather. Because there is no analog in weather observation. That means you can't simply record the patterns, since we started recording patterns of metrics of the atmosphere, pressure, temperature, wind, etc. And by using pattern recognition of the historical past, know pretty accurately what's about to occur. It's that lack of an analog that required computers in the 1940s to begin to integrate models, equations of motion that actually make some sense in the atmosphere. And so I think this has bearing on how we plan what we do. In all of the effort of folks and everybody in the room, do we see any evidence of strong enough analogs to think we can rely on? And in some parts of the brain, there are. I mean, if you're looking at bird songs, it's great. You can be perfectly 100% predicted by matching little patterns of the start of a bird song to know everything that's about to occur and even what bird it might be. But in seizures, I think looking at the literature, we may have problems in that. That also means that collecting more data, bigger computers, might not lead us to what we're all interested in. So I'll throw that out as a basic question. Do you think there are analogs? That is, there's periodicity in the dynamics of the system enough to let you use what I understand about pattern recognition?
Steve, I'll use that as an opportunity to kind of take umbrage of one of the things you said, which was that you kind of don't care about either mechanism very strongly in terms of efforts, or even which signals you measure. Because I think in a lot of ways there are traps. There are traps in the way that we've collected data. There are traps in the ways that we've gone about the signal processing and the machine learning methods to try and do prediction that disenable us from being able to both model and understand what may be going on inside the brain, and what mechanisms may be leading and modulating the overall seizure susceptibility. I mean, we know that, for example, behavioral state, you mentioned, sleep states and other things that make that, in a correlation basis, certainly seem to modulate the seizure susceptibility. And that kind of data goes back forever. We saw work reported from the Gowers, and then blanking on the names from the 1930s. Tracking the cycles of sleep, and if you look in detail at that early work where people were really strongly regimented in their sleep behavior in the epilepsy colonies, they were woken at 6 in the morning, and so you could see really strong driving by their driven sleep states, and you know that going in. You can then abstract back as to what the sleep states were that the people were in, which were driving their high susceptibilities. But to get back to the questions that Steve was asking, in the early 1900s, when we were trying to begin building weather prediction, the measurements that were being made of pressure and other things were the underlying measurements of the hydrodynamics of the Earth's atmosphere. And if we're not measuring the right things, right? If we're not measuring the sleep state, and the sleep state is regulated by parts of the brain that overall regulate the awareness level, the excitability level of different parts of cortex, then how can you say that you're going to be able to predict the susceptibility to transition into seizure if those things are inherently modulating that ability to transition out of stability into seizure? Or what's this effect of that leads into susceptibility to spreading depression? And is there a balance between them, as the paper that you referenced of Steve's seems to imply? And how often are spreading depression events observed in, or actually occurring in, freely behaving people, who are having epilepsy? Are they coming out of their seizures? Are they leading into their seizures in places that we're not measuring? Because nobody makes those measurements. Anyway.
One could even be a bit more provocative with these black box models and say, if we use them, it's like throwing mud at the problem and see what sticks. And over the years, I guess we threw so much mud at the wall that a lot of things stick. And then we cleaned the wall and call this statistics. But the statistics is obviously also limited. And when you again start throwing the same thing, some will stick at least for a while. And that might even make its way into clinical trials. And you might even win one of those at some point. The question is, what do we have at the end? Do we really have something that predicts seizures? Or do we just won the lottery? Being a millionaire because I got it right, and it turns out years later that it was a random process and it didn't keep to its promises. After a while, that was really tested.
I think so. And if you want to intervene, can you intervene without knowing what the underlying mechanisms that are kicking it into instability are? Or, I don't know.
Hilda.
What I'm going to say may sound like advertising and a bit hypocritical. So, yesterday, I presented a poster session, a few people came, but not everybody, and in which we have, most of the things you'll be hearing here of prediction, they require that the patient be in a hospital all the time. And so yesterday we presented an idea of the device which we are starting to be slowly but which is portable. So the person doesn't have to have a big thing of electrodes on the head. But the most important thing is that it's run by an algorithm which is able to see the precisions where they are. And it's able also to distinguish between a pre-seizure and a real seizure. And so in a way, I don't know if I can say that it's hope, I'm not a medical doctor, I'm a physicist, so that's where I come with something like that. And what we saw was that we have analyzed a lot of data, and what we saw is that you have alerts. I mean, with every precision that you measure, you can send an alert, and then the alerts have a maximum. The first alert, because there may be precision, the first alert has a maximum, although they run up to an hour or more in the data that I have. So, but they have a maximum around 25 minutes before decision. And the important thing, as I say, is that is something that can run only with a couple of electrodes that you can sort of stick. Hopefully, everything will be tested on people, and we are going to do it in the hospital in Sao Paulo. But the important thing is that a person will be able to carry normal life. To carry on a normal life with a very small thing that is not more aggressive than a hearing aid, and will be able to send signals to whoever can help. I mean, there is a possibility of recession. So, well, as I say, it sounded like has no
First question, of course, is does it work?
Sorry?
The first question, of course, is does it work? Because there have been a lot of studies and a lot of trials where people were wearing the device. The Melbourne is, I guess, the best example. It's even an implanted device.
Yeah, but of course, I mean, that's what I said. The algorithm on which we've been working for a long time, for some time, and we have tried it in a lot of data in which I know that there is a seizure, and that's how I started looking for the possibility of having signatures of that seizure coming. And I can tell you that in the data it works in about 90% of the cases, because the others essentially did not have any precision. And we are going to start with the hospital, but we cannot try. We have not yet done the real gadget. We are on the process of making the gadget. So the device is more nice. So, but it will be very small, and then we will try to get the hospital to help us with the patients that will carry it, and then we'll see. Is as I say, it's a project.
I understand that it works. You just said when there is a seizure. Does it also work when there is no seizure?
Sorry?
Does it also work when there is no seizure? I understood that you said...
You are right on that. The thing is, in the data, I have looked in ictal and interictal data, so I can tell you that whenever there is something that may sound like it, in the interictal region, and it's not, it's different. So I don't know if whether it will be telling you that there is a seizure coming, and there isn't, until we make the real measurements with real people. I won't be able to tell. So, but for the moment, it's a project that seems to work very well every time that I had done that. And the first type of work that we will be doing with the people from the hospital everyone knows, is the advertised in Sao Paulo. And we will work on that particular point that you are saying with the data that they already have, but they won't tell me, well, this is there. So it's nothing like when I collect other types of data that you can easily find around, in which it tells you there is a seizure here, and when I had the idea that there was something before, that's what I did. I collected one when the seizure, and then I started looking backwards to see if there were signals or not. Because I mean, you see that kind of thing in many other problems in physics.
Okay,
So
we might continue that conversation outside with her. Look, she's obviously got a lot of practical problems about doing the study. Greg's got a question over there.
And just through the panel, so give us your best, what else we should be measuring. Local fields, units, I think, probably pursued less, but certainly local field potentials, you know, pretty exhaustively. What's the signal that holds promise?
Before we try to answer that, because I don't have an answer, I would like to add another question. If you tell us what signals we should measure and what else we should measure, how do we combine this? Because we have no mathematical tools, no theory that would allow us to integrate all sorts of wearable devices. As you mentioned, it's maybe a great idea, but we don't have the mathematics. We don't have any techniques at the moment that would allow us to combine these various sources other than downsample everything to, I don't know, the temporal resolution of the slowest thing or whatever. But this whole theory is basically missing.
But I do think that we are measuring the signals that we have to measure. The thing is that we are not making sense of the data that we are getting. And we are not making sense of the data because there are so many axes of variability that we are mixing up that it's impossible to extract meaningful information. So that is why I try to bring the point that we need to order how we collect and we organize the data. Now, if you come from the research, animal research field, you know that you cannot record local field potential from freely moving rats and use whatever signal at whatever time you like. You know that you have to first make a clear state definition. You need to try to focus in a particular behavioral state of the animal, and then try to focus in a particular signature of the local field potential in order to connect with the questions that you want to answer, which typically are related with the mechanisms. So now, I am an expert in trying to understand the mechanisms of whatever signal you give to me, and try to tell you what were the single cells doing during that particular event. I can do that, but I can do that if you give me the correct data from the human. I can do that. I mean, me, basic researcher. We are professionals in doing that, but I do need the correct signals. So my point is that if we make a serious effort in trying to bring a clear standard to the field, to the clinical research field or data collection field, as long as we can, because I understand that at the end of the day, you have a patient that you have to treat of a problem. But as soon as you can do that, we will be able to organize better that. So it's not the same, again, to be recording from the core, from the penumbra, from a region which is not directly related. We need to know the connectivity in order to make sense of why is increasing something here that is decreasing in other patients. In a poster yesterday that were analyzing these interictal spikes for hundreds of seizures and organized by patient, you see that there is a circadian effect. But each patient was different. And we know that it's not one gene, one disease. So we know more and more that there is so much variability. The axes of variability are so enormous that we need to make sense of that, to try to make groups. And this will help us to make sense of the data. Just to finish, fast ripples were described by someone who come from the basic research standards. And the first things Anatole Bragin did was to open the filter band and to record with microelectrodes, tiny electrodes which were minimally invasive, and open the filter band. This never happened in clinics because you are forced for other circumstances. I mean, you have what you have. But this opened a whole field and show you that now HFO can be used. And now we need to understand what is an HFO in a macroelectrode versus a microelectrode, and what is normal and what is not. That's the point. So we find the answers if we organize the data collection and we try to learn and to connect with the mechanisms, because they will be informative. That's the point.
Can I add a different perspective? So, like Bjoern, I don't know what to measure, but I'm tempted to say that we should try to measure things that we at least think are associated with seizures being more likely to happen. So there are things that patients tell us. For example, people with epilepsy often say stress. So we don't really measure anything that actually directly measures stress-related phenomena. And we know seizures might occur in particular sleep stages, and we know that seizures are much more likely to occur if you've recently had a seizure. So, there are things that we don't really measure because we focus so much on the electrophysiological data, but, we should be measuring patterns of seizure occurrences, things related to stress, things related to sleep stages, I think.
I think Martha was next.
So there was a comment before that we need to know about the mechanisms, but let me propose actually that most of the therapies that we have for epilepsy were discovered empirically. And that when we discovered that certain things work, we look back and actually learn something about some epilepsy. And so I turn to that I would reinforce the comment that we're dealing with a very heterogeneous disorder, at least in humans, and this makes it more challenging. But if we look at the Sante study and then at the studies from the RNS system where people have been stimulated all over, and the results are very encouraging. They're not perfect. There are some people who become seizure-free. There are others who do not. And we can learn a great deal, I think, from those who are seizure-free or who are doing very well and those who are not responsive, and go back. Because we can talk about the prediction, but perhaps just going ahead and treating people, and then understanding why there is a response or not a response is as favorable. And I'll just make one last point, that when we started working on the RNS system, I thought very naively, this was 16 years ago, that we would be intervening. Patient had 3, 5, 6 seizures a month, clinical seizures a month, and we would be intervening about that often. And then we discovered, of course, that when you put these electrodes in people's brain and they walk around, it's astonishing how ill they are, and how really very non-revealing clinical seizures are as far as the burden of their disease. So the whole concept of stopping seizures, I think, is not relevant any longer. It's neuromodulation. But the question is, when do you intervene? When do you modulate? And now most of the patients going through the RNS system are having intrarectal epileptiform discharges that are typical of those that sometimes precede a seizure. So the average patient will be getting 600 or 2,000 stimulations a day for about 100 milliseconds. So about 5 minutes altogether a day. So this data, of course, is very rich. But I would suggest that you don't necessarily need to begin by understanding the mechanisms to go ahead and intervene therapeutically.
Marty, I've got 2 questions for you. One is this idea that you say that, I'm not sure exactly how you phrased it, these patients are more sick than we expected. It's kind of do you think that all of these interictal discharges have cognitive impact on them? And is there a sense when you're treating them for them that's intervening in that cognitive impact?
I can answer that indirectly, and this manuscript was just accepted in Epilepsy, and David Bjoern is the first author. But there was detailed neuropsychological testing done. In the mesial temporal patients, there are statistically significant improvements in memory that are not correlated with changes in the clinical seizure count. And in patients with frontal lobe epilepsy, there are statistically significant improvements in a number of aspects of language that, again, do not directly correlate with the clinical seizure count. And when one looks at what might be biomarkers, which would be electrographic seizures that we all would agree are electrographic seizures. Mark Quigg recently looked at this data and found there was wide agreement amongst observers and what is an electrographic seizure. And then also interictal abnormalities. When one looks at that, what happens with those is if those go down, the patient's clinical seizure reports do not necessarily, and vice versa. But when you look at these electrographic seizures, you have to be convinced that the seizure diaries are so inaccurate. And in fact, as people get better, they may start to be able to count more seizures. And that certainly, certainly see that to be the case, that as seizures become less severe, they're more reliably reported.
Right. And so another question I have, maybe actually putting back out to the membership here, which is we talk about seizure prediction as if we could predict the seizure, it's going to happen based upon some measurement, right, or some current state. And this question comes up, episodically all throughout the history of this meeting. Are seizures really deterministically going to happen, or are there states at which there's a heightened probability or heightened susceptibility to seizures, and the state of the brain comes up to but doesn't always reach the actual seizure, and then goes back down to lower susceptibility. And so if we ever succeed very well at measuring whatever that susceptibility is, we will never get perfect prediction, because in that case, the probability is that susceptibility is only a measure of susceptibility. It's not a deterministic going to have the next seizure right now.
May I ask? This is what I think is needed. I mean, this is what we need. We need to know where the optimal place is to put the electrode, whether that is at the seizure focus or whether that is in propagation pathways. And that's complicated because there are so many regions where seizures may arise, and these people's anatomy is not normal. It's not normal in everybody in different ways. So we can't assume that we understand propagation pathways in one individual. But the other, so that's where to put the electrodes, and the other is what type of electrographic activity should be interrupted, and what's the goal, what's the state that should be achieved. Because the technology to any squiggles, any lines can be measured, can be detected. Now the engineers will tell you that that's very simple. We just have to tell them what we want to detect and then where we want things to be.
Okay, look, we've got to a really fascinating part of the discussion, but we are a bit over time, so I might just conclude on Liset's comment to that.
Just a short point regarding that question that you ask. You are asking for the mechanism. When you ask us where to put the electrode, what you implicitly ask for is what are the mechanisms. Now, just very briefly, we are so many, and we are so different, and we think so differently. We have different brains here, thinking on a problem, that we don't need to choose what side of the room we want to take to have a look to the whole room. So we can look at the mechanisms, and we can look at having some practical perspective, and then to try to make common language to understand. That's the successful way to go. So we don't need to choose whether doing rational drug discovery or serendipity. Just let's work together and try to make a common language, because that's the point. The point is that the answers are in the same room. It's just different perspective.
Okay, look, thanks so much, panel. It was a very stimulating discussion, and the audience. A great session. Thank you.